Now we are going to do plasmid extraction using the conventional alkaline lysis method. Plasmids are extrachromosomal DNA which may contain antibiotic resistance genes. In recombinant DNA technology, plasmids are used as vectors to clone a gene of interest which may express or produce proteins with biotechnological applications. Plasmids are usually transformed into E. coli competent cells, grown in LB broth media amended with antibiotics. Positive transformants expressing the antibiotic resistance genes in the plasmid can thus be easily screened from an agar medium containing the antibiotics. The objectives of this activity are to describe the conventional method of plasmid extraction and to explain the principle of alkaline lysis method. The materials include bacterial culture in broth media, 1.5 ml microcentrifuge tubes or Eppendorf tubes, microcentrifuge tube racks, refrigerated centrifuge, micropipettes, pipette tips, 50 ml conical tubes, conical tube rack, ice bucket, and gauze. Collect a single positive colony and grow in 250 ml LB broth or TB broth medium with antibiotics for 8 hours. There are 7 steps in plasmid extraction using the alkaline lysis method. 1. Cell collection. 2. Cell resuspension. 3. Alkaline lysis. 4. Neutralization. 5. Plasmid precipitation. 6. Washing of plasmid. And 7. Elution of the extracted plasmid. For step 1, transfer the bacterial culture in 50 ml conical tubes and centrifuge at 12,000 rpm for 5 minutes. Carefully discard the supernatant. For step 2, resuspend bacterial cells in 10 ml of solution 1 containing trees, HCl, EDTA, RNase, and glucose. Resuspend completely by pipetting up and down. Incubate at 37 degrees Celsius or room temperature for 5 minutes. EDTA chelates divalent ions like magnesium and calcium, essential for DNA's activity, thereby protecting the plasmids. For step 3, add 20 ml solution 2 containing sodium hydroxide and SDS. Gently mix by inverting the tube several times. Sodium dodecyl sulfate or SDS is a detergent which denatures cell membranes and most proteins. Sodium hydroxide disrupts the hydrogen bonding in the double-stranded gDNA and plasmid, which are converted into single-stranded DNA or ssDNA. Place the tubes on ice for 10 minutes. Add 15 ml solution 3 containing potassium acetate with glacial acetic acid. Gently mix by inversion. Potassium acetate facilitates the precipitation of SDS protein complex. Glacial acetic acid allows for the renaturation of single-stranded plasmid into double-stranded DNA, which easily dissolves in the solution, while the single-stranded gDNA forms complex with the denatured cellular proteins through hydrophobic interactions and forms white precipitate. Place the tube on ice for 10 minutes. Then centrifuge at 12,000 rpm for 30 minutes. For step 5, Filter supernatant using gauze while transferring into a new tube. Add 27 ml isopropanol, mix gently by inversion. Then centrifuge at 12,000 rpm for 20 minutes. Discard the supernatant. For step 6, add 5 ml 70% ethanol. Resuspend the pellet completely by shaking the tube. Centrifuge at 12,000 rpm for 5 minutes. Then discard the supernatant. For the final step, add 1 ml of tea vapor. Resuspend the pellet completely by pipetting up and down. Tea vapor dissolves the DNA and protects it from degradation by DNase or RNase. Store the plasmid DNA in -20 or -80 degrees Celsius. This result is from my PhD thesis showing extracted plasmid DNA from recombinant bacterial cells. The extracted plasmid DNAs are showing 3 distinct bands which correspond to the 3 types of plasmid DNA: the nick plasmid at the top, linear DNA plasmid at the middle, and supercoiled plasmid at the bottom. Supercoiled plasmids are highly intact circular plasmid. This plasmid extraction having thick bands of the supercoiled plasmids is said to be of high quality. These can also be evaluated using the actual plasmid DNA quality measurement using nanodrop, showing an average DNA quality of 1.82.